Shin is a team leader from Riken Center for Integrated Medical Sciences. He's co-leading international consortiums such as Phantom 6 and Human Cell Atlas. Jay enjoys building functional genomics platforms to decipher the molecular mechanisms involved in gene regulation, cellular plasticity, and disease.
Okay, good morning, everyone, and good evening, good afternoon from other parts of the world. Thank you, Goji, for introducing and also for putting this wonderful virtual meeting together. And it's a great pleasure to introduce to you about the Human Cell Atlas Asia efforts. And as you have heard this fantastic talk from Aviv just now, there has been a tremendous effort globally to build this Human Cell Atlas. So the question is, what can the Asian community do, and how can we forge a collaboration that can also not only represent, but also lead in areas that can really contribute to the global issues such as disease and COVID-19. So today I'm going to kind of give you a past history of what we have been doing and what we hope to do in the future, and hopefully that this can rally up some of the community members in Asia to communicate and to collaborate and to forge new research ideas. So let me see if I can change this slide here. Okay, so as you can see, as you have seen also from Aviv, there's a fundamental mission that we all abide by and really want to make sure that we can create this comprehensive reference map across the human genome. Across the human body. And one of the ways to do this, of course, is through collection of multiple cell types from multiple locations. And one of the key ideas that really stood out to me when I first joined the Human Cell Atlas is that there are key principles that we really want to abide by, and that really is the community and diversity aspect of the Human Cell Atlas. And this quotation is just straight from the HCA white paper, which is that cells will be studied using broad range of techniques to capture both breadth and depth that will fully represent the world's diversity. But furthermore, the Atlas is the HCA community is committed to genetic and geographic diversity and equity at every phase. So inspired by these missions, we really wanted to make sure that we can also come together as scientific communities in Asia to represent and fulfill such missions to cover and to fulfill the completeness of the Human Cell Atlas. So I'm kind of going to introduce to you, many of you are familiar as we are in Asia, but I think this is a good reminder that there's a lot of interesting, abundant genetic, ethnic, cultural, even environmental and socioeconomic diversity in Asia, because we are home to about 60% of the world's population, where the two most populous countries like China and India are in Asia. I think in contrast to Europe or the United States, there's a bit of a challenge that there's lack of central government that really incentivizes the scientists in the region to come together to collaborate on. And that's really kind of one of the key missions of the HCA Asia, is to break down barriers and being able to find common interests to bring research that can benefit the societal needs of the region. And this is kind of represented in this graph here, in that if you look at the global genomic data, that only 1 or 2% of the genome is of Asian ancestry. And this really limits the clinical utility of how we can use genomic data for Asian population. And this is another study from GTEx, where, of course, it's a US study, but only 1.3% or so is representative of Asian descent. So this naturally, invariably contributes to the biases in clinical data, genomic data, which will have a negative or unexpected outcomes for clinical implications. So with that backdrop, in 2017, Piero Caninci and myself actually organized a first Human Cell Atlas Asia meeting in Okinawa, Japan. And there the purpose was very clear: to meet, discuss about what are the latest science and technologies of single-cell genomics that are happening in the region, and to identify what are some of the societal needs and challenges and how we can overcome them, but also to strategize increased participation, representation, and leadership in Asia. And as you can see in the picture in the bottom left, it was a rather small group. It was about 40 members or so, but it was very intense, focused discussion that have led to very good network of collaborations that in 2018, Woon-Young Park and Sung-Young Kim decided to organize the second HCA Asia meeting in Jeju, South Korea. And just last year in November, Shayan Pravakar organized a third HCA Asia meeting, which had about 300 participants to really discuss these issues of how to bring science and technology in the region together. So I just want to give you some takeaways from these meetings and how this have led to our current standing and how it's going to, we hope to see pan forward. So we noticed from all of these discussions in these meetings that there are very interesting and active single-cell research that are happening with many shared interests. And I just want to kind of give you some of the highlights of the research that are currently ongoing in various countries in Asia, going by different countries. But hopefully by end of these highlights that we can find areas that could come together. So for example, in Japan, there's a lot of efforts in technology developments, such as Ramda-seq or single-cell cage-seq, but also three-dimensional imaging called Cubic that has been very powerful to build a atlas based on tissue clearing technologies. There are unique samples also in Japan, such as supercentenarians, which will be presented by Piero later in the symposium, but also iPS technologies and how it can be used for regenerative medicine, and creating a correct cell annotation for that is one of the key programs that is really important in the country. But also later in the symposium, you'll also hear talks about how we can use organoids to study COVID-19 research. In South Korea, they've been active in Human Cell Atlas from the beginning. And they, as I mentioned earlier, they hosted our second HCA Asia meeting in 2018. And because of their dedication and support for the Human Cell Atlas, they were actually able to formulate their own group within South Korea, which they have their own steering committee, and was able to reach out to their local governments to acquire single-cell program funding, which led to six projects involving proteomics, spatial, longitudinal studies, and cancer and immune disease. That also extensive colorectal cancer, lung cancer. And I think recently, Woon-Young Park and others are further exploring the research of COVID-19 patients using single-cell technologies. I think Singapore has also been very active in single-cell research, and I will not go through every single one of them, but their involvement in cancer has been very impactful, as well as the immune systems, studying the inflammatory dendritic cells, as well as brain macrophage developments. But also computationally building algorithms have been very big impact from Singapore. I think it is also important to highlight that the hosts of this meeting, the China, have also had tremendous efforts and contributions in single-cell research. I think in 2019, the scientists in China has gathered to kick off this primate cell decoding project called PCDP, and it's currently led by Sunyi Zi, Wenzi Ji, Zhu Zhang, and Jiaqun Wang. And all of these efforts are really to decode the genetic regulation in primate cells using single-cell technologies. They've also come together to build atlas of blood cells, and that's led by Tao Chang and others. And it's really to aim to build a reference map for all hematopoietic cells. And I think it is selected as top 10 medical advances in China in 2018. Again, as we all know, GoJiGo has been pioneering this cell landscape projects, and is aiming to construct cell landscape for major species. And this really shows how genome encodes the cell landscape using artificial intelligence. And so far, they've created more than 3 million cells that have profiled across human, mouse, zebrafish, and Drosophila, and others. I think China has also been leading in the front of technology development, and I think there are many ways to utilize and contribute to the Global Genome Cell Atlas as well as HCA Asia efforts. So I just wanted to kind of summarize that, yes, there are active single cell research that are ongoing with many shared interests, such as immunology and cancer, and that there are unique technologies and methods that can really be broadly distributed and shared for highly standardized and harmonized approaches. And I think it's also important to note that in Asia, there's tremendous genetic and environmental diversity, that we have access to unique disease cohorts, which again, in return, have high clinical utility of the data set. But I think we could also be aware of the challenges that we are facing in the region. And that is that there are varying local regulations, such as ethics and data release policies. And how we need to overcome this requires a constant dialogue, not only among scientists, but also with policymakers and also the public awareness. And the second challenge is that many funders, especially in Japan and others, are shifting towards more translational disease studies rather than basic science or technology development. So we hope that we need to make sure some of our research are meeting the needs of the funders, as well as advancing in basic research. The third challenge is that we do face in Asia is that 29 out of 49 countries in Asia are considered emerging and developing countries. So this will touch upon the issue of equity and being able to reach out to these nations to support and train the local scientists in order to contribute their scientific knowledge into the HCA efforts. And the fourth challenge that we recognize is, as I mentioned earlier, the lack of central government or the funding body really makes it challenging to incentivize scientists to build communities. And in return, we also have decentralized tissue biobanking platforms, which also makes the data standardization or sample coordination a little bit more challenging. So whenever there are challenges, we believe there are opportunities. So we believe we need to, it's an opportunity for HCA Asia community members to come together to build harmonization of data and standardization of the workflows, and really to identify common interest projects that can benefit not only a single country, but the whole region and the population in the region. So one of the ways to do that, of course, is through organization of these meetings, to have discussions and breakout sessions to talk about these common themes and research ideas. And in the meantime, provide opportunities to promote equity, gender balance, and ethics awareness to the region, so that some of the data and scientists are equally represented. And hopefully through this, we can create opportunities to co-apply or support local and international fundings. So just to go over this quickly, to facilitate a lot of these efforts, we are in the process of creating a HCA Asia steering committee. And really the value of that committee and the main mission of that committee is to build a community in Asia to enhance the communication with local scientists. And that we can also use opportunities to showcase regional science and strengthen regional research alliance, and also to identify synergies, and in the process, diversify the Genome Cell Atlas program. So we will the committee will constantly work with the global HCA to coordinate these annual meetings and to collaborate with also equity working groups to provide the right communication aids and ethical toolkits. And we will build communities through identifying key research themes that can benefit the region. So here are just 5 examples, such as cancer, diabetes, infectious disease, aging, and genetic diversity. We really hope to rally the scientists in the region to touch upon these research topics to expand and collaborate. And hopefully we can use that opportunity to co-apply for both local and international funding, and that the data will be contributed to the Human Cell Atlas. And I believe that all of these efforts will help us to achieve this equity and diversity and reach the high clinical benefits of the genomics data and meet the local societal needs. So I'm just going to go through really quickly these 5 flagship projects because from tomorrow, actually, there's a breakout session, and we will really hope to engage with the participants of this meeting to discuss how we can build communities around these 5 themes. So one of them is called cancer, and I believe cancer is a global issue, but there are very specific concerns in Asian region that speaks to the local communities. And here's just an example of a colon and rectum cancer that are highly prevalent in Japan or Singapore, but conversely, we have uterine and cervix cancer, which are more prevalent in Cambodia and India. So just recognizing and identifying these concerns and issues that are prevalent in different parts of Asia, and being able to provide the right reference data set for the healthy, but also providing the right toolkits to generate single cell data will be critical. I think diabetes is one of the key important issues in Asia. I think there's a bit rapid growth of diabetic cases in Asia compared to any other ethnic backgrounds. And that could be partly due to changes in diet and lifestyle. However, there are key indications of genetic contributions that may have contributed to diabetes onsets, as well as resistance to certain diabetic treatments. Here in the Department of Health and Human Services in the US, they state that about 40% of Asian Americans are more likely to be diagnosed with diabetes than non-Hispanic ones. So again, there's even in the same environmental factors, there's a higher risk of Asians for diabetes, and something that can really come together as a community in Asia to tackle this issue. Third is infectious disease, and we all recognize this through recent times, but also there are local disease burdens and local pathogen strains that just differ from many parts of the world. And due to the lack of central governance in Asia, it really makes it challenging to standardize a lot of the efforts to address some of these infectious disease. So hopefully we'll have an opportunity to talk about what are the areas and hurdles that we can overcome to address this. Aging is, I say here, is a disease of the present and future. As you can see, the highest ranking cause of disease is Alzheimer's disease in Japan and South Korea, or age-related neurodegeneration, compared to 2007, where it wasn't the case. So there's a growing number of aging population in developed countries, and we envisage that in the future, this will most likely be the case in many other countries in the world, including in Asia. So preemptively addressing some of these aging questions would also help the communities to come together and tackle some of these challenges. The fifth item that we would love to discuss tomorrow is genetic variation, as a work by the GenomeAsia 100K Consortium. And there are about 10 different ancestral groups and lineages in Asia, which have a tremendous impact on the human disease and pathology. Therefore, just understanding the genetic basis across the population in Asia were critical in diagnosis and prognosis of disease, and even the treatments of particular drugs for the treatment of disease. So understanding the genetic variation as a backdrop in the context of disease will be extremely critical. And to partly address some of these issues, some of us in the Human Cell Atlas Asia committee have initiated a group called Asian Immune Diversity Atlas. And this is a project, a pilot project that are currently funded by Chan Zuckerberg Initiative. And Chung Cha-Hon and Kami-san and Jay, Shin, myself at Weekend is working really closely with Eun-Young Park in Samsung, Korea, and Shayan Prawakar in A-Star, Singapore. And the goal really is to profile the human peripheral blood mononuclear cells across multiple individuals. Here, we're aiming by 500 individuals and using multiplexing strategy that we will hope to understand how the genetic variations attributes to gene expression patterns across the population in Asia. So we have built up a very strong network of community and we have a Slack workspace, we have Google Drive to exchange files, and we are successfully multiplexing 16 individuals into single 10x runs and generating a very cost-effective profile of the local population and the immune cells. Here is just a snapshot of integration of human PBMCs from 3 different sites, Japan, Korea, Singapore, that we're successfully integrating them. And we are also profiling their genotype in order to understand genetic variations and how that attributes to gene expression. So I just want to acknowledge CCI again, and thanks to their efforts, we are now expanding AIDA, which will include Thailand as well as India. And hopefully we can further expand these efforts across Asia, including China and Vietnam and other parts around the world. So if you are ever interested in joining AIDA, please do let us know. So I know we kind of talked very high level of what we have been doing and what we want to do. I just hope that it painted a picture of what are some of the areas that we can come together to work on and to address some of the challenges that we are facing. So hopefully in the next, today and tomorrow, we can identify what are the strengths of the HCA Asia community and the scientists, and how we can utilize their know-hows and techniques to build further collaborations in the region. Hopefully, we can also find common needs through flagship projects, and through the steering committee, we'll be happy to organize and help to facilitate a lot of these discussions and scientists to formulate these efficient flagship projects going forward. So I just want to remind you again that feel free to join us tomorrow at day 2 breakout session number 3, that we will kind of touch upon these issues in more detail to put ideas into action. So hopefully your participation would be greatly appreciated. So I just want to close by thanks to Shyam and Eun-Young for a lot of their inputs into this, and Yoshinari Ando and Piero Caninci, who really brought the HCA Asia concept to fruition, and CCI for funding a lot of the projects that I've talked about today. So with that, thank you everyone, and hope to talk to you again later tomorrow.
Thanks, Jay. Very good talk. And we have a question on the Slido from Julia Zhu. For difficult genetic analysis of diseases like ASD, what's the advantage of using ATLAS model compared with conventional methods?
Yeah, I mean, let me try to read the question again. Yeah, I think the ATLAS model comes in 2 different dimensions. So of course, ATLAS, as we would been talking about, as well as Aviv has mentioned, is it provides a vast cellular diversity and the cellular context. So cell-to-cell interactions or spatial contexts, just understanding how neurons or Schwann cells or other particular cells are interacting, and what are their diversity in their interactions and how they communicate. Just having that context, the microenvironment provides a very rich understanding of any disease, where conventionally looking at bulk tissues were averaged or focusing on particular cell type. So hopefully the concept of Atlas will bring in this resolution of cellular diversity and heterogeneity that will answer some of the challenges that facing disease. But the second component, I think, which you're referring to is genetic analysis. Again, there are attributions of how much genetic diversity can influence the gene expressions. And I think this is a very much ongoing discussion of how we can understand genetic contributions to gene expression. And one of the examples that have been pioneered by GTEx is, how can the genetic variation in non-coding regions, such as in the cis regulatory elements, enhancers or key transcription factor binding sites, have been really understudied in the field of genetic research, largely because they were just very difficult to associate them to a particular disease or gene expression. Many GWAS studies have identified SNPs in the non-coding regions, but people have been focusing on exons or protein coding regions. So this is really an opportunity to understand how we can attribute these non-coding regulatory regions to gene expression, and how that can attribute to the makeup of a cell or the function. And I think we do need genetic diversity or the genetic landscape to understand this. So that's one of the efforts of the AIDA project, as I mentioned, is how the genetic diversity attributes to gene expression. Hopefully that can be a jumpstart to many other tissue cell types that's already included in the human cell atlas.
Great. Now we have another question from Hongming Zhang. Might you share your experience on single cell meta-analysis across different races?
Yeah, that's a really good question. We are exploring that as we go as well. How do you define ethnicity? And that is something that we want to stay unbiased as possible. So for example, in the AIDA project, when we collect samples from various individuals, we do ask their ethnic background. Although those are optional to answer, these are kind of personal interpretations, so to speak, of their background. So it is a difficult and somewhat sensitive topic. So one of the ways that we are trying to stay unbiased is to use their genotype information. So that is using just SNPs arrays, we are able to profile their genotypes and correlating that to the gene expression. So yeah, it's an ongoing discussion. I'd be happy to discuss how we can do this more systematically. But at the moment, we are relying more on the genotype profile.
All right. Because of time, we have a final question from Alistair Forrest. How does HCA Asia handle mapping to different genotypes, population-specific genome alignments?
Yeah, so this, I don't know if I could answer that question properly, but yes, obviously different ethnic backgrounds have different genotype profiles as well. As I mentioned in the AIDA project, we are using the SNPs arrays, the genotype arrays that are specifically designed for Asian population. So this is something that we are mapping our genotype to. How to broadly apply this concept to different regions around the world needs to be further discussed. So yeah, I don't know if I'm answering this question appropriately to what Alistair is asking here, but we are using a predefined set of SNPs that's catered to Asian populations for genotyping at the moment.
Thanks, Jay.